you all understood about the definitions of enzyme inhibitors. Now you all have to understand about the classifications of enzyme inhibitors. So for understanding the classification of enzyme inhibitors, you have to understand that this is having the two types. Okay, so the first one is your reversible inhibitor, and second one is your irreversible inhibitors. So if we are talking about the first one, that is reversible inhibitor, so they cannot bound with the enzyme with strongest bond, means they are bind with the help of non-covalent interactions. They cannot share electrons with enzyme. That's why those bond which they are sharing with enzymes are weakest bond. Okay, so if they are sharing the weakest bond with the enzyme, definitely they are not able to produce any kind of chemical reactions. And as these bonds are so weaker, so it is easily possible that you can remove that kind of bond with the help of dilutions. Okay, that's why it is known as reversible. You can reverse this. That's why it is known as reversible inhibitor. The example of this kind of bond is hydrogen bond. Now if we are talking about the subtypes of reversible inhibitor, so the first one you have to understand is competitive inhibitors. Okay, so competitive inhibitors are those which are competing with the substrate. Okay, in place of substrate, they bind with the enzyme and they will not allow the substrate to bind with the enzyme. That's why it is known as competitive. They are having the competition with the substrate. The second one is uncompetitive. Why it is known as uncompetitive? Because these inhibitors bind only to substrate enzyme complex, means they will firstly allow the substrate to bind with that particular enzyme, and after that when the substrate enzyme complex will be formed, they will bind with that particular substrate enzyme complex. That's why it is known as uncompetitive inhibitor. The third one is the non-competitive inhibitors. Why it is known as non-competitive? Because binding of the inhibitor to enzyme reduces its activity but does not affect the binding of substrate. Means they are not affecting the binding of substrate with the enzyme, and but they will affect the activity of enzyme. They can reduce the activity of enzyme. That's why it is known as non-competitive inhibitor. The fourth one is your mixed inhibitor. Why it is known as mixed inhibitors? Because it is having the competitive and non-competitive both kind of nature. How? First one, inhibitors can bind to the enzyme at the same time as substrate. Okay, so they are wanting to bind at the same time when the substrate is wanting to be bound with the enzyme. Means they are having the competitive nature against the substrate. That's why they are behaving like the competitive inhibitor. But the second thing is the action is vice versa. The action is vice versa means the substrate can also compete with that particular kind of inhibitor. Okay, so it can also affect the binding of inhibitor. So that's why it is known as mixed inhibitors, because the action is vice versa. Now the next one is your irreversible inhibitor. Okay, so for understanding the irreversible inhibitors, you all have to understand that reversible means which can be reversed easily, and irreversible means those which cannot be reversible easily. That's why it is having the covalent bond, means they will share the electron with each other and they are having the strongest bond as compared to the reversible inhibitor. Okay, so they cannot be reversed with the help of any kind of hydrolysis. And second point is, in this the enzyme substrate complex is formed, means they are allowing the enzyme to bind with the substrate, and after the enzyme substrate complex formed, they will bind with the enzyme substrate complex. So they are generally the non-competitive kind of inhibitors. So hope you all understood everything. Thank you so much.